Good afternoon, Year 12. So, today we're going to carry on with the DNA, genes and chromosomes topic by working through a lesson on mutations. Now, this is a particularly important lesson when it comes to understanding things like cancer and other genetic conditions, and so it's a very important one for anybody who's interested in going on to do further genetics or any form of genetic medicine. Now, there's 3 questions on the board which should have been covered last lesson through translation. Have a glance at those questions. If you could pause me and just write the answers, not the full question, just the answer, somewhere on your paper. So, you've got 3 questions there. So the first one is by far the longest one. Describe the basic stages of translation in full. And what you're looking at there is you're looking at, firstly, the mRNA leaving the nucleus. Once it's left the nucleus, of course, it will be spliced, but we're not going to include that as part of translation at this point. It will move towards and attach to a ribosome. The codon on the mRNA will attach to the anticodon on the tRNA, and each tRNA will contain a specific amino acid. Therefore, the order of the codons will determine the order of the amino acids. Each amino acid will be joined to the next amino acid by a peptide bond, and the ribosome will move along the mRNA until eventually you reach a stop codon. Very difficult to say what the individual marks will be there, but as long as you're writing down all that information, then you can have no issues at all. What does the term degenerate mean with reference to genetic code? What that means is that there's more than one code for each amino acid type. So AAA and AAU and AAC might all code for the same amino acid. And again, remember, there's no need for you to have memorized which codons lead to which amino acids. But degenerate means there's more than one code per amino acid, and that becomes important today. And then the third question, what does the term universal mean when you refer to the genetic code? It means that all organisms that we are aware of have exactly the same genetic code, i.e. CCC in bacteria will code for the same amino acid as CCC in a human or a pig or a bird or a tree. So after all that, let's move on to today's lesson. Today, as I said, the lesson topic is mutations. That's your title, is mutations. And we're going to be looking at different types of gene mutation. We're talking about what gene mutations are and what causes them. And we're going to talk about what a couple of different types of mutations are, including deletion and substitution mutations. And at the end of that, we're then going to describe what a mutagenic agent is and identify some possible mutagenic agents. And this is all on the syllabus. These are all things you need to know for your A-levels. This will become very relevant when you go on to do some work on cancer with your other teacher. You may already have done that, in which case you can apply this to that lesson. So let's get started. Firstly, the first question we have to understand is, what is a mutation? And we should have done this at GCSE, so I'm hoping that most people will be able to give me a good answer to this. And hopefully what you're thinking is that a mutation is a change, a base change in the genetic code, in the DNA. So a mutation simply means that somewhere in your DNA, a base that was, let's say, an adenine, has changed into a thymine or a cytosine or a guanine. So one of your bases, or more than one of your bases, has changed. Now there are lots of different kinds of mutation, and we're going to study 2 of them today, and you'll come across 2 more in Year 13. I'm going to start with the really simple, easy ones today, though. I'm going to start with addition and substitution mutations. Before that then, and incidentally, of course, there's your note. If you don't know that is what a mutation is, that you should write that down immediately. The question then is, how will that affect the organism? And there's 3 possible options here, isn't there? You could have a positive effect, you could have a negative effect, or you could have a neutral effect, i.e. it doesn't really make much difference. If it's positive or negative, that will be because we've changed the amino acid that we were using in our protein. So we've changed the base, which has changed the mRNA, which has then changed the codon, which has changed the anticodon, which has changed the amino acid we're going to use. So now instead of a lysine, let's say we have a tryptophan. That will only make any difference if it then changes the shape of an important protein. The tertiary structure. Now, I'm going to be playing you a short video about this topic. It's a really nice simple one. In all honesty, this is quite a simple lesson. So what I'm going to ask you to do again, I'm going to play the video on your screen. Again, if that isn't working, the YouTube link will be directly underneath, and I will try to remember to put it in the link descriptions for this video underneath it in YouTube.
DNA rarely makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by 1. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by 1. Again, the resulting protein will likely be non-functional. Lastly, an inversion will invert a segment of DNA end to end. Watch this section closely. Each base has been moved around. Again, these mutations have the potential to be harmful, but not all mutations have an effect on protein. Remember, during translation, a start codon must first be read to begin coding the protein. If the mutation happens before or after the protein coding sequence, there will be no effect on the protein. Mutations must occur in the coding section of DNA to affect protein synthesis. Otherwise, nothing happens. Thanks for watching this episode of Teacher's Pet.
So I'm going to stick some questions on your screen. What I need you to do is to pause me and to have a go at these questions. The questions should be coming up there. There you go. Now, if you don't remember any of these answers, just go back and re-watch the last section of the video or watch the video directly on YouTube. But have a go at these questions. So, hopefully you were able to follow that. And it's quite a nice simple video, that one. It is worth keeping that one back because it is quite useful. It did include one type of mutation we're not going to talk about today, and we're actually going to leave until year 13, but there's no harm in having learned about it. So the different types of mutations that she talked to us about were additions, where we add one base, deletions, where we remove one base, substitution, where we're going to change one base for another, and then she talked about the inversion mutation, where we're going to effectively flip the DNA around 180 degrees so that it's in the wrong order. Now the only ones we mind about for today are the substitution, the addition, and the deletion, and those ones we're going to focus on in a lot of detail. The next question we're going to talk about is why some are more dangerous than others. Now there's two things she talked about there that are relevant that you may have got down. The first is talking about something called a frame shift. Now a frame shift is the idea that if you add a base in or take a base away, you don't just change that one codon, you change every other codon after it. So if you can imagine, I would usually draw this on the board, but if you can imagine a series of 3 base codons. So let's say your mRNA said AAA, TTT, CCC, GGG. That was your mRNA. 3 A's, 3 T's, 3 C's, 3 G's. Fine. Let's say that we added a T in right at the beginning. So now instead of saying AAA, TTT, CCC, GGG, now it says T, AAA, TTT, CCC, GGG. It doesn't just change that first base. What's going to happen is that the first codon is now TAA, the second codon is now ATT, the third codon is now TCC, and so on. So it's changed every codon after it. And these are called frame shift mutations. And again, this can be quite a complicated thing to get your head around. The reason I chose that video is because I think it does show it very effectively. If you don't understand what I'm saying, do go back and just pick that little bit which talked about frame shifts. Have a look at it again. Then we come to talking about the second reason why some might be more dangerous than others, and that is effectively the answer to the third question. The idea that mutations are only really dangerous if they occur in a part of the DNA that actually codes for a protein. So she talked about it happening before the start of a gene. That could happen. Let us say that the mutation happens before the gene even starts. Well, then it doesn't matter, the gene hasn't been affected. But we know some other parts of DNA that don't lead to proteins being created. We know about introns, and you may remember about introns from splicing, I hope you do. Introns are those bits of DNA that we cut out during splicing because they're not used to make proteins. If a mutation happens in an intron, a lot of the time it won't make much difference. Not always, but a lot of the time. I'm going to look at a couple of examples in a minute. So, let's look at a couple of examples. Now, the two main types of mutation which we need to be aware of are deletions, and therefore, by substitution, by process of knowing what the word addition means, additions as well. So I expect you to know what an addition is and a deletion. And then substitution. Substitution is the other type of mutation. So by the end of the day, deletion, addition, substitution. So deletion is we delete a base from the code. Substitution, we change one base for another. And those are the only types of mutation I need you to know about by the end of this lesson. Every other kind of mutation, and there are lots, will be covered by year 13. So please pause me and take these notes down. So, deletion and substitution. And let's see if we've understood this. What I'd like you to do is I'd like you to have a look at the questions on the board. So, this is a section of coding DNA, and it's got set sections. Now, I would like you to do the questions that are there. Now, there will be a coding table in the assignment to help you do number 6, or just use the coding table you downloaded last time for the translation lesson. So have a look at that, have a go at each of those questions, and pause me now. So, what will be the pre-mRNA made from this DNA? Well, you've got to make it complementary to the DNA. So we are looking at UACAGCCCUAAGCCCUG. Fine. The final mRNA, well, what's the difference going to be? We're looking at removing the introns during splicing. That's the difference. So it's going to be shorter, and it's not going to include that intron. So now it will just be UAC, AG, AGCCC, UG. How would a substitution mutation to a C of the red base affect the protein translated? Well, if you think about it, that red base is just in the intron. So if I substituted that for a C, it shouldn't make any difference at all, because I'm going to delete that bit of DNA effectively when I make my final mRNA, before I make my protein. So that shouldn't make much difference at all. If I added another base after that red base, that might have an effect. And that's effectively question 4. Well, how would a deletion of the green base affect the protein? Well, what's going to happen is every base afterwards is going to shift along by 1 back towards the C. So now it will just say, the DNA will say ATG, TCG, GG, ATTC, and it will skip straight to the GGAC. So we effectively just remove that base entirely. What would the final mRNA be? Well, again, to get bored of me reading out letters, but we're looking at UAC, AG, AG, CC, UG. So we just remove a C. And then you're going to use your coding tables to work out which of the amino acids we're going to. So if I make one pop up, this is the one that you should have had. So we're looking at the final mRNA for the first piece of DNA before we have the mutation. So we're looking at a UAC for a start, which would be tyrosine, followed by an AGA, which should be a arginine, ARG. Then we're looking at GCC, so we're looking at an alanine. And then we are looking at a CUG, which is going to be leucine. So before we have the mutations, as far as I can see, that should be the amino acids. If we have a mutation where we delete that green base, we're going to be still looking at it being identical up to that base. So we're still going to have UAC, which will be, as I said earlier, tyrosine. Then we're looking at a AGG, which we're looking for a arginine. But then we're going to skip straight on to the GGA there, which should be a CCU. So that will now instead be a proline. We're not going to have the final amino acid, so we're going to end up with a shortened version of the protein, which is going to be completely different at the end. Okay. So the next bit we're going to talk about is why most mutations will have no effect on the final polypeptide created. We already talked about one reason, the reason she gave in her video. And that reason was that actually if it happens before the DNA even, before the gene even starts, then it'll make no difference. We also talked that if it happens in the intron, it will make no difference either. A lot of the time. Can you think of any other reasons why most mutations will have no effect on the final polypeptide created? Well, I'll give you a hint. If you don't remember any, we're thinking about the word degenerate here. So, if we go back to our coding table, if you think about it, if I change from a UCU on the mRNA to a UCC or a UCA or a UCG, I still get serine. So often, if you have a mutation, actually you will end up with exactly the same amino acid. And then the final reason, which I wouldn't necessarily have thought that you would expect you to think of right now, the final reason is that even if you change the amino acid, often that will have no effect at all. A lot of amino acids are quite similar. There are positive amino acids, negative amino acids, there are aromatic and non-aromatic amino acids, and all kinds. But a lot of the time, amino acids with similar codes will have similar properties. So I would think that phenylalanine, top left, would have the same rough properties as leucine, which is directly underneath that and has similar codes. That's not guaranteed, and it certainly won't be the same, exactly the same, but if you only change one amino acid and it's only a very slight change and it has similar properties, that still won't lead to a change in the tertiary structure. So we've effectively got 4 reasons. 1, it might not even happen in the gene. 2, it might happen in an intron and therefore might not make any effect. 3, the code is degenerate, so even if you do change an amino acid in the exons, it might not have an effect to what a base in the exons, it might not have an effect on what the amino acid is. And 4, a lot of amino acids have similar properties, and so it might not make an effect even if you do change an amino acid. Remember, there are thousands of amino acids per protein, and so changing one often will make no effect. Fine. Now, there is an exam question. What I would like you to do is I would like you to complete this in full. Now, we are changing slightly the way we are working from home. I'm no longer expecting you to upload all of your work. I'm going to ask you to upload specific, important bits of work. So this exam question is the only bit of work I want you to upload from this lesson. You can write it in your book. I'd like it written out in full. You don't need the question, just the answers. But I'd like you to write it out in full. And I'd like it photoed and sent to me via Teams, please. Or you can do a Word document. So pause me. This is a full 8 marker. That should be 2 marks at the bottom. Now, what I'd like you to do is I'd like you to send that to me, but I am going to give you the answers here because I think actually it'll be more useful to you. I will still want to look through this. I want to see where people are struggling with this question. So, explain how this mutation can result in the production of a non-functional protein receptor is the main and most important bit here. So, the first bit is it will change the DNA base. That's just knowing what a mutation is. You get a mark for that. And you could talk about deletions, substitutions, or additions if you wanted to. The second mark is saying that it will change an amino acid, will change the primary structure, which is effectively the same thing. The third mark is for suggesting that that might alter the position of various bonds. The fourth mark is for saying that that will change the tertiary structure of the receptor. So why would having a mutation lead to a non-functional receptor? Because firstly, that will change the DNA, that's what a mutation is. Secondly, that could change the amino acid. Thirdly, that amino acid change might affect the type of bonds you get in this protein. And fourthly, that might therefore change the tertiary structure of that protein, making it non-functional. People with CCR5 show a greater resistance to developing AIDS. Explain why. Well, this is all from what you should have done on AIDS earlier in the year. And we're looking at the receptor not being complementary to the HIV, and therefore the virus not being able to replicate. And then finally, the frequency of the CCR5 mutation is highest in Europe. Scientists have collected data on the history and number of HIV infections in Europe. Now, why do we think that the CCR5 mutation is not due to natural selection in response to HIV? Well, there's several possible reasons. 1, few people have HIV in Europe, relatively. 2, HIV hasn't been around for long enough for natural selection to occur. 3, mutation has been around for a long time. And 4, therefore it must be the mutation is advantageous for some other reason. The first 3 are the ones I would expect you to be writing. Now this is common sense, maybe not. This is not something I'm going to be teaching you. A lot of the time with these questions there's going to be a 2 marker at the end which just gets you to think about it. Now the only bit of the question that I personally care about is part A here. Because this is what we just talked about, particularly the first 2 marks, and down here, 3 and 4, is from what we talked about in protein structure back in the biochemistry. It's worth noting, things do come up again and again. So if you don't understand something in one topic, it's very important that you get help and assistance with that, or work through it yourself, because often it will come up again next topic. So if someone didn't understand protein structure last topic, that will hold them up in this topic, and it will probably hold them up in topics to come as well. So if you don't get what tertiary structure is, contact me, I will happily help you. Okay, now that's the end of that. From today, all I need you to know is what a gene mutation is, and and how it arises, what is meant by a deletion and substitution mutation, interpreting base sequences to identify gene mutations and their impact, which is effectively what we did here. And this is the kind of thing you will see in the exam, though a little bit less fancy, no colours. And then this last one, actually, to be fair, I'm going to spend just a minute just talking about this last one. It's not difficult. Mutagenic agents. Broadly, mutagenic agents are any factor that will cause a mutation. So that could be a chemical, or it could be radiation, it could be lots of things. So a mutagenic agent, it might be worth just taking this down as a one-sentence note. A mutagenic agent is any factor that will cause a mutation. Identify some possible mutagenic agents. Again, this is common sense, things we think of when we think of things that will cause mutations, cause genetic diseases. The main ones we want to be thinking about are radiation, particularly with things like skin cancer, ultraviolet radiation, but a lot of forms of radiation will cause mutations. And then we've got chemical mutagens, things like smoking, amongst a lot of others, but smoking is kind of the classic one. Okay. If you've got all three of those, and the last one, again, it's a little bit of common sense, but hopefully you'll understand that. If you've got all of those, then you've understood today's lesson, hopefully quite a short one, because it's not too difficult. If you come across other types of mutations in your book, we will cover them in Year 13. They're Year 13 topics. These are the only ones you need to know for Paper 1, which is the Year 12 material. Okay, have a great day, Year 12, and I look forward to seeing you whenever I get to.